When we look inside of a skeletal muscle fiber, we mostly are seeing myofilaments, the proteins for contraction. Don't forget that the myofilaments are bundled into groups called myofibrils. So on the picture here we can see the skeletal muscle fiber with its sarcolemma. Inside of that we have bundles that are the myofibrils, and then within the myofibrils we find the proteins for contraction, the myofilaments. There are two types of myofilaments, thin filaments and thick filaments. The thin filaments are thinner and the thick filaments are thicker. In the picture here up at the top, the green are the thin filaments and then the purple are the thick filaments. When you look towards the bottom of the screen, the thin and the thick doesn't come off as well because they haven't drawn them to scale as well. The thick filament is made of just actually one type of protein called myosin, and it's made up of a whole bunch of myosins bundled together. Myosin kind of looks like you took two golf clubs, twisted their shafts together, and then you have the two heads sticking out at the one end. And we call the heads, and then we call the shaft the tail. To form the thick filament, the tails kind of get glued together, and then the heads stick out on the outside. The function of the thick filament, the function of myosin, is going to be the fact that it pulls on the thin filament. So the thick filament does the pulling during muscle contraction. And this process will require ATP. We talk more about that in another video. In the little GIF at the bottom of the screen, we can see that thing that kind of looks like the golf club at the top, and that is the myosin that's pulling on the thin filament. And again, the thin and thick filament in that GIF are not drawn to scale, which is why the thin filament looks thicker than the thick filament. The thin filament, even though it is thinner, it's actually made of three different types of proteins. It's made of actin, tropomyosin, and troponin. The main component to the thin filament is the actin. So in these images here, you see the light green spheres with the little dark green blotches on them. So those light green spheres are the actin proteins. The little splotches are little binding sites on the actin where the myosin is going to grab onto. The actin isn't just like one long string. It's actually two long strings twisted around each other, like you took two strands of pearls and then twisted them. Tropomyosin is sort of a rope-like protein. In the picture here, it's shown in orange and it looks like it's like a long rope. Tropomyosin has the job of sitting on top of those binding sites on the actin, and essentially the tropomyosin hides that binding site unless something happens to reveal it, which we talk about later. Sitting on top of the tropomyosin is the third and last protein of the thin filament, and this is called troponin. And a lot of the pictures, the troponin looks sort of like a snowman or a dog's Kong toy, if you've seen one of those before. Troponin has the job of holding the tropomyosin in place. The only way the tropomyosin will ever move is if troponin itself moves. The function of the thin filament overall is that it will get pulled by the thick filament. So the thick filament does the pulling and the thin filament is what gets pulled. And this only can happen in the presence of calcium. Calcium will bind to troponin, causing the troponin to move a little bit. In biology, when one thing binds to another thing, it causes a little tiny bit of movement. So when calcium binds troponin, that snowman, right, the snowman moves a little bit. And the troponin is attached to tropomyosin. So if the troponin moves, the tropomyosin will also move. And when that tropomyosin moves, those little dark green splotches, those binding sites will become revealed. And don't forget, the myosin will bind to the actin at those binding sites. Then after it binds, the myosin will pull on the actin, and that's going to be our contraction event. So as the thin filament moves, the muscle is also, the whole entire big muscle is also going to move, which I find really, really awesome, how we can take these teeny tiny proteins that we can't even see with a microscope because they're so small. but somehow they're going to move our whole entire muscle, right? It's amazing. In this picture here, we can see what the thin filament looks like when it's active. And a thin filament is active in the presence of calcium. So that calcium binds to the troponin, causing the troponin to shift, and that will shift the attached tropomyosin, revealing the binding sites on actin. In the presence of calcium, the myosin of the thick filament can grab onto the actin. And when the thick filament is holding onto the actin, we say a cross bridge has formed, because you could take a little molecular person and walk them across the myosin over to the actin, right? There's a bridge now between the actin and the myosin. In the presence of ATP, that myosin head is going to be able to pivot, pulling on the actin. The myosin will then let go and grab onto another actin and pull, and then it will let go and grab onto another actin and pull, and it keeps doing this over and over again. Sort of like we see in the GIF image here of the person pulling the rope, right? You have to let go so that you can grab another part and pull again. Thick and thin filaments are organized into groups called sarcomeres. Each myofibril is composed of multiple sarcomeres lined up end on end on end on end. So in the picture here, we can see the multiple sarcomeres making up the myofibrils. Sarcomeres are made of highly organized, overlapping thick and thin filaments. Because the thick and thin filaments are laid down in such an organized way within the sarcomere, it creates that dark light pattern. It's dark where the thick and thin filaments overlap, and it's lighter where there's no overlap. And this produces the dark light pattern that we call striations. Within the sarcomere, the thick filament is going to pull on the thin filament. In other words, myosin will pull on actin. And as this occurs, the thin filament will slide past the thick filament. And this is what we call the sliding filament theory of muscle contraction. Don't forget, in science, theory means something of which we're most sure. Theory often means something very different outside of science, right? We often think of theory as something that has no evidence, like a conspiracy theory. In science, though, a theory has lots and lots and lots of evidence. So we have a lot of evidence that shows us that during muscle contraction, the thin filament slides past the thick filament. When this occurs, the sarcomere will become shorter. So at the bottom of the screen here, we can see how during the contraction process, the sarcomeres become shorter, and during relaxation, the sarcomeres become longer. In this picture, we can see the difference between a relaxed and contracted sarcomere. Notice the length of the relaxed sarcomere and how much shorter the contracted sarcomere is. And again, what's going to cause the contraction process is the fact that the thin filaments are going to move past the thick filaments. If you look at these two pictures, you can see that the thick filaments are in the exact same position. They haven't really moved their position in the sarcomere at all. However, the thin filaments have been pulled towards the center of the sarcomere. And by pulling the thin filaments towards the center of the sarcomere, you're causing that sarcomere to shorten. We can't finish our talk about thin filaments without talking about something called costomeres. And this is a point of confusion for students, so you might want to star it, underline it, highlight it, whatever you want. Costomeres are actually proteins in the sarcolemma. So these are a collection of proteins in the cell membrane of the muscle fiber. What does that have to do with the thin filament, right? Well, these proteins in the sarcolemma are attached to the thin filament, so that when the thin filament moves, these costomere proteins are forced to move. And because these costomere proteins are embedded in the sarcolemma, that forces the sarcolemma to move. Additionally, these costomere proteins are attached to that endomysium, that connective tissue membrane that's just outside of the sarcolemma, so that when the thin filament moves, the costomeres move, the sarcolemma moves, and the endomysium moves. Now don't forget, the endomysium, like all of the other connective tissue membranes of the muscle, the endomysium merges to form the tendon. So if the endomysium moves, the tendon will move, and the tendon is attached to a bone, so if the tendon moves, the bone will move. And what I love about Costomeres is that it connects that microscopic stuff that's happening inside the cell, that moving of the thin filament, to show us how, in the big picture, moving that thin filament is going to end up moving our bones.